All right, so this is our first note section about membranes. A lot of this stuff is review, so it should be pretty easy. It's also in your book, so there's some good stuff in there too. So we'll go through this. There's a lot of slides, but I put in some pictures, so it won't be as bad as it looks. So every cell is surrounded by a plasma membrane. Sometimes in your book it'll be called a cell membrane. Same thing, just different name for it. There is a little difference, but don't worry about it. Plasma membrane regulates the passage of things in and out of the cell. And in eukaryotic cells, there's actually membranes on the inside of the cell. Remember, there's membrane-bound organelles. So that's the endomembrane system right there. We talked about what it's made out of. It's made out of phospholipids. We learned about those in the chemistry section. We learned about amphipathic, what that means. Right? That means that they have a hydrophobic section and a hydrophilic section. So one part of it likes water and the other part doesn't like water. The part that doesn't like the water is called the hydrophobic tail of the phospholipid. The hydrophilic part is the head. Remember, it looks something like this, something like that, a whole bunch of them. Where this part right here likes water, this part right here doesn't like water. No water. Yay, water. Right, so amphipathic, and that's what that means. Here's a picture of it, a little better picture than the one I drew. Here you can see the phospholipid bilayer. There's two layers. There's two layers of phospholipids with their tails towards each other, because these all inside here are hydrophobic. Right, that's hydrophobic. Out here is hydrophilic, which is good because this is mostly water out here and in here. This is made up of mostly water. So it's a good thing that these are hydrophilic out here on the heads, and that the tails on the inside are the ones that are hydrophobic. So the biolayer is it's called a biolayer because there's two layers. Should make sense. The two hydrophobic layers are the ones that are next to each other. Right? So they really like each other, but they don't like water. The shape, having a round head, makes them all kind of fit together and associate with water better. That's pretty much all that one says. Here's a picture of it. You can see, we'll talk about this picture or another picture like it again. There's lots of stuff going on in a membrane. You can see all the phospholipids here, right? That's all these blue things. And there's their tails in here. And then inside here you can see things like cholesterols. Remember we talked about cholesterols. That was a type of lipid also that was made had 4 rings, right? That's a lipid also, a cholesterol. We know that. There's lots of proteins. There's those proteins there. There's that protein there. These long skinny ones are also proteins. Right? So there's lots and lots of proteins stuck in this cell membrane. Besides having phospholipids and cholesterol, there's lots of stuff in there. This part right here is called a it's a sugar molecule, so it makes it a glycoprotein, and we'll talk about that very soon, probably. So this whole membrane, the theory of this membrane, the model they use is called the fluid mosaic model. Oops, mosaic, not mosaic, sorry. Erase. This thing is so cool. Mosaic model. Right, so it's fluid, means it flows and it moves. And how fluid it is or how rigid it is depends upon what it's made out of and what kind of lipids are on the inside of it. Now, most of you guys know that when things are cold, they don't move as well as when they're hot. So when the outside temperature of a cell gets cold, they'll change the content of their membranes to make it stay more fluid and not get too rigid. Cholesterol is one of the big things that helps maintain fluidity, especially in cold temperatures. And that's why we need to have cholesterol. We can't have too much cholesterol, right? Otherwise it clogs our arteries, and that's not good. But we need to have some cholesterol. We can't have no cholesterol. Their lipid bilayers are flexible, so that means they can move and bend without breaking, and that's important because these are holding our cells together, so we don't want them breaking. They also, if they do break, they're self-sealing, so they can kind of fill back in again if there is a hole there. They kind of fit back together. They also make their own little vesicles. So if we have a cell bilayer right here, and it kind of buds off a little bit. So if we have it, oops, I don't want to do that one. Let me erase that. It can kind of make its own little bud like this, right, to put some stuff in there and make its own vesicle. Eventually it'll go back together and we'll have this kind of a vesicle there, right? So it's able to make its own little vesicles and seal itself back up. Now the proteins inside there, they're integral proteins. Those are the ones that are firmly bound to the membrane. Some of them go all the way through the membrane. Those are called transmembrane proteins. There's some that are stuck on the outside of them, and we'll show you the picture. They don't go all the way through, but they're stuck into it just a little bit, or on the outside of it. They're stuck on there. But they're integral. They're stuck there. They're not going anywhere. Here's some pictures. So here they kind of ran out of room, but you can see right here, this one that doesn't go all the way through, that's a peripheral one. This one right here that goes all the way through, that's an integral protein, or a transmembrane protein, because this goes all the way through. And here, the ones with these extra sugars on them are called glycoproteins. These are called glycoproteins right here, because they have they're proteins with the sugar on them. And that's what this talks about right here. It says that glycoproteins are proteins that have sugars added, and they're made right in the ER and the Golgi complex, and they get transported out. Exocytosis is something we'll talk about later. That's stuff leaving the cell. That's stuff leaving the cell. So when something leaves the cell, the outside layer of the vesicle becomes the inside layer of the cell, and you'll see that later. Don't worry about that so much right now. Cell membrane and permeability, how stuff gets through. So it's called a selectively permeable. It means it lets some stuff through but not others, and that's what we're about right now. That's what I just said. Allows some things through but not all. Some substances can get in and some can't. At different times it may block a particular substance, but then later allow that because it needs it. Right? So there's lots of different things that can be going on in the cell. And by doing that, the membrane in the cell can help control how big it is, its volume, and the internal conditions, what it's made out of, what kind of ions it has, and what other kind of molecules are in it. So it can help control internal conditions, which, I should go back, but I'll write it up here, which you guys remember is called homeostasis. Can't forget that. That's one of our favorite words ever, homeostasis. So usually the things that are most permeable, the things that can go through the most, are small nonpolar molecules, which means they're hydrophobic, like oxygen and carbon dioxide, small ones, right? Oxygen is O2, carbon dioxide is CO2, so they're really small. Water can usually go through, because it's small enough to pass through the gaps. What can't go through are charged ions and large molecules, things that are large and polar, can't get through there. So those are stuck on the outside, and that's something that's really impermeable to Earth. Here's a diagram that helps you see that. So anything that's small, hydrophobic, anything that's nonpolar, or water can usually go in and out pretty freely the cell through the membrane. Now, for those things that can't get through normally, we have transport proteins. These are proteins that help move things across, including ions, amino acids, sugars, and other big molecules that are polar that need to get through. There's two basic types. There's carrier ones and there's channel ones. And they're just exactly the way they sound. Carrier ones need to carry the thing across. Channel is a gate pretty much where things just flow right through. A channel. In each type of transport, protein transports a specific type of molecule. There's very few that are just, hey, everybody come in. It's usually a specific thing that it allows through. So carrier transports or proteins, those are the ones that bind the ion or molecule. So they bind it and then they change shape to move it across the membrane. There's two forms. There's facilitated diffusion and carrier-mediated active transport. Lots of words. We'll usually just call it active transport, and we'll talk about that later. Facilitated diffusion is just regular old diffusion that we'll talk about in a minute, but it's helped by these proteins. Active transport is different because it needs energy, and we'll talk about that when we get to that. I think that's in the next section, actually, not this section. It needs energy for it to transfer. That's why it's called active. And channel proteins. Channel proteins are just pores. Like the pores in your skin are holes that oil can come in and out, or dirt can come in, and bacteria can get in, and you can get zits. But pores are holes usually, and that's what channel proteins are. So they just kind of open, and they have gates. They can open and close, and then they allow stuff to come in and out. Picture here, I think. Here you go. So here's a channel, a protein channel. You can see it's just open. Stuff can go through. You can go this way if it wants to, whatever. The carrier proteins actually grab onto it, right? Change shape and then spit it out the other side. So they do kind of the same thing, but they're just a little bit different. So passive transport. This one has to do with simple diffusion and facilitated diffusion. We kind of just talked about that. That's so easy. So should be pretty easy. Osmosis is a really good one. Everybody's seen Osmosis Jones, but this is different. This is still osmosis, but not Jones. So we're going to look at some of the stuff to do with those diffusion things. The first thing is, gotta learn this. This is really important. Concentration gradient. So we know what concentration is, right? That's the amount of stuff in a certain volume. And remember, we talked about it being in something called moles per liter last time. A gradient is how it changes. If something's graded, it's going from high to low. If you go up a steep grade, it's a really steep hill. If you go down a steep grade, it's a really steep hill. A shallower grade or a lower grade is not such a steep hill. So concentration gradient is a change in concentration. So if we have a whole bunch of things over here, and like one or two over here, right? The concentration is much greater over here, so there's a concentration gradient. That's the gradient. There's a difference. High concentration, low concentration. That's the gradient. Diffusion is the movement of stuff down the concentration gradient. Things want to go from high to low. So if there's a whole bunch of molecules over here, right, and there's none over here, all these molecules are going to want to move over here and spread out. They don't want to be, well, they don't really want, but they're not going to want to be right next to each other. They're going to be one nice and spread out. So they go from high concentration to low. Here's some pictures of that. So here's some water with some dye molecules to make it be colored water. Maybe it's red or yellow because it looks kind of yellowish, right? There's lots of dye molecules over here, very concentrated. None over here, so low concentrated. High concentration, this is high concentration side, this is a low concentration. That's a concentration gradient. And we'll see in a minute that what happens is the stuff wants to flow this way. So it happens that this membrane allows the dye to go through until finally it's the same on both sides. Now it's a lot lighter than it was over here, right? Because if this was a dye, this would be a really dark color. And now this would be a little bit lighter, right? But the same on both sides. So that's what we're going to look at. If a membrane is permeable to a substance, then there's a net movement from high concentration to the other side, and it kind of performs energy. It's able to do work because of this. Those molecules want to go over there, and it actually is going to make energy. And when we get to photosynthesis and cellular respiration, you're going to really see how important that is, how this gradient of having a whole bunch of stuff on one side and nothing on the other side is going to create some energy and be good for our cells. When there's more than one type of different substance in there in a mixture, they all diffuse independently of each other. So I hope I have the next picture up there, but that one picture had, let's do it like this. Let's say we have circles over here, right, a whole bunch of circles over here, and X's over here. The circles are going to want to be diffused this way, right, and the X's will want to be diffused that way, independent of how much stuff there is. So having things move back and forth, it doesn't matter what's on the other side. They're just worried about themselves. Once everything's all balanced out, it's called in dynamic equilibrium. That doesn't mean things stop moving. That just means they start moving at the same rate back and forth. So if we have a membrane here and we have 3 X's over here and 3 X's over here, there's a good chance that this X might move over there and this one might move over there, but it's going to stay at about the same thing. It's possible we'll have 4 over here for a while and 2 over there. That happens. But in equilibrium, it will balance back out again as long as it's forth freely. So here's that picture. I knew I put it in here. I couldn't remember that for sure. So these red ones over here are going to want to diffuse this way, and these yellow ones are going to want to diffuse this way until there's the same amount of both on either side, and that's when it's in equilibrium. A simple diffusion is just stuff moving right through the membrane. Down the concentration gradient, that's important. It has to go from high concentration to low concentration, but simple diffusion is just stuff diffusing right through the membrane. Right? No help, simple stuff. Osmosis is kind of the same, except now it's water moving. So it's water moving through a membrane instead of the molecules. Right? Since a lot of the solutes, there are things that are dissolved. Remember, solute is the thing that's dissolved, and water is usually the solvent. That's the thing that did the dissolving. So things are usually dissolved in water. These things are usually too big to go back and forth. Then the water moves to change the concentration so that they're the same. Remember, it's always concentration. Concentration, concentration, concentration. Not the number of stuff, but the concentration. The amount of it per amount of water. And that's what it is. So here's a picture of some osmosis happening. These are sugar molecules in a water separated by a semi-permeable membrane. It's semi-permeable that the water can go back and forth, but the sugar molecules can't. So as you can see, there's a lot more sugar molecules on this side, so this is a high concentration. This is low concentration over here. Now, the sugar molecules want to go over here. These sugar molecules really want to go over here really, really bad, but they can't. They can't get through there. But what can happen is the water molecules, H2O molecules, can go that way. And it'll actually raise the level of this until the concentrations are the same. So there's still a lot more sugar molecules over here, but there's a lot more water now too, and that balances out and makes the concentrations are equal. So until the concentrations are equal. Like I said, not equal numbers of things, equal concentrations. Looks like an A. Yeah, good enough. So osmotic pressure, now the pressure of osmosis, it's the amount of pressure that would keep that water from rising. Right? It's the amount of pressure you'd have to push down on that water to keep it from flowing over there. And that's what osmotic pressure is. And it can actually get pretty high if the conditions are bad enough. So if I had the osmotic pressure of this is the amount of pressure it would take for me to push this down so that this whole thing looked like this. That's what osmotic pressure is, right? Because this water wants to move over here, and it's not going to listen to us. It's going to do it no matter what. If we push it down, we have to add force, which creates pressure, and then that keeps that. There's different types of solutions because of this. It's called tonicity, which has to do. You'll see it as usually something tonic. So isotonic, iso means the same. That means that there's no net movement of water on both sides, so that the cell is in perfect, it's like an equilibrium. When it's isotonic, everything's in equilibrium, and they're happy. When there's a higher concentration of dissolved substances, if the outside is more higher concentrated than inside the cell, the fluid is hypertonic to the cell. So if we have a cell right here and there's lots and lots of stuff out here, change the color actually, change to lots and lots of stuff out of here, lots and lots of stuff, and not a lot in here, the water of the cell, the water is going to want to go out. Water goes out. It leaves the cell, and it causes the cell to lose water and to shrink. If the opposite happens, if inside the cell there's lots and lots of stuff, right? If there's lots and lots of stuff inside the cell, looks like a sad face. That's funny. Oops. It's hard to make dots with this thing. But not so many outside the cell. The water's going to want to go inside the cell. So water's going to want to go in. It's going to cause the cell to swell up, possibly burst, possibly blow up, or lyse, and we'll talk about that in a minute. But that's hypotonic. So Here's a better picture than I can draw, obviously. Here's normal. There's the same amount of water going in as there is out. If it's a hypotonic solution, there's lots of water wanting to go in here because that water wants to balance the charge, and eventually it's going to blow up the cell. These happen to be blood cells. This happens to our blood. If it's in a hypertonic solution, all the water leaves, and we get a shriveled up cell. In plants, they're a little bit different because if you remember, plants have that cell membrane there that helps protect them. So what happens is if they have if the water is going into the cell, right, if they're in a hypotonic solution, they're going to swell up, but they get really firm and really rigid, or the real name is turgid. It's turgidity. If it's the opposite, if it's hypertonic and water's leaving the plant cell, the cell wall still kind of keeps its shape a little bit, but you can see the cell membrane starts to pull away from the cell wall. And becomes turgid pressure is how turgid it is, right? It's the water pressure pushing against that cell wall. So it only happens when it's in a hypotonic solution. So all that water moves into the cell through osmosis, building up the pressure against the cell wall. Right? It's very, very important in plants that don't have wood because it gives them support, like trees so much. If a cell that has a cell wall is placed in hypertonic solution, the cell shrinks, and when the membrane separates from the cell wall, it's called plasmolysis. Plasmolysis is the cell membrane shrinking away from the cell. And here you can see those again. So here's the turgid part, and here's the plasmolysis. Facilitated diffusion is pretty much simple diffusion, except now something's facilitating it, something's helping it move along, and that's the facilitated part. And those are proteins. So some type of transfer protein, whether it's a carrier or a channel, will help it move. Again, it always moves from higher to lower concentration, and it could be a channel or a carrier protein. And here's some pictures, or a picture. So look at 1, 2, 3, 4, 5, 6. 1, 2, much higher concentration, lower concentration. So everything's going to move this way through here. Easy stuff. That's the last slide. I'll have another one for the second part with talk about active transport when we need energy to make things move across, and we'll do that one next time. All right. Good job.